Hello and welcome to Edu Aspirants, our YouTube channel for competitive exams. Continuing with our series of videos on cell, the fundamental unit of life, this is third and final video, cell organelles and functions. Structural organization of cell. A typical cell consists of three major components: plasma membrane, also known as cell membrane, nucleus, and cytoplasm. Plasma membrane or cell membrane. Structure. It is the outermost covering of the cell. It is about 6 to 10 nanometer thick layer mainly composed of phospholipid layers and proteins. Functions. It is also known as selectively permeable membrane or SPM that allows only certain type of substances to pass through it. It protects the cell from external environment. It is because of the flexibility of plasma membrane, certain organisms like amoeba are able to engulf food material by the process called endocytosis. Nucleus. Structure. It is a double membrane structure mainly consisting of nuclear membrane and chromosomes. Nuclear membrane. Nucleus is surrounded by a double-walled nuclear membrane. It has pores at certain places called nuclear pores which help in exchange of materials between nucleus and cytoplasm. Chromosomes. Chromosomes are thick rod-like structures which are visible at the time of cell division. During non-division activity, they remain diffused as chromatin material. Chromatin consists of DNA and proteins. DNA is the hereditary molecule. Functions of nucleus. Nucleus is the center of genetic information, so it has command and control on all the activities inside a cell. Nucleus provides information for protein synthesis. Nucleus plays a crucial role in cell division. Nucleus determines the way a cell will develop. This is done by directing and controlling metabolic activities. Cell organelles. The cytoplasm contains various cell organelles such as mitochondria, endoplasmic reticulum, Golgi bodies, lysosomes, etc. Various functions of cell depends upon the activity of these organelles. Endoplasmic reticulum. Structure. It is a large network of membrane-bound tubes and sheets. Structure of ER membrane is almost similar to that of a plasma membrane. Types. Depending upon the presence and absence of ribosomes, ER is of two types: RER, that is rough endoplasmic reticulum, and SER, that is smooth endoplasmic reticulum. Functions. It helps in transport of materials within the cell. It functions as a cytoplasmic framework to provide a surface for various metabolic activities. RER helps in protein synthesis while SER helps in synthesis of fat molecules. In liver cells, SER plays a crucial role in detoxification of poisons and drugs. Synthesis of cell membranes with the help of proteins and lipids by endoplasmic reticulum is called membrane biogenesis. Golgi apparatus or Golgi body. It was first discovered by Camillo Golgi in 1898 and also known as dictyosome. Structure. It consists of many membrane-bound vesicles and tubules which are almost parallel to each other, forming stacks called cisterns. Tubules are often connected to ER membrane. There are two distinct phases of Golgi. The concave phase is called transphase and convex phase is called cis phase. Functions: assembling and packaging. Various substances synthesized by ER and ribosomes are assembled in Golgi. These are packaged and dispatched to various targets through Golgi bodies. Formation of lysosomes. Golgi bodies are also involved in synthesis of lysosomes and other membranous structures within the cell. Complex sugar formation. In some cases, complex sugars may be made from simple sugars in Golgi apparatus. Lysosomes. Structure. Lysosomes are single-walled spherical muscles which contain very strong hydrolytic or digestive enzymes. Functions. The main function of lysosome is digestion of materials that enter the cell. They are also known as digestive bags. Lysosomes keep the cell clean by destroying worn out cell organelles and other foreign materials. Sometimes, during disturbance of cellular metabolism, the lysosomes may burst and damage the cell parts, which may cause death of cell. So, lysosomes are also known as suicide bags of the cell. Mitochondria. Structure. They are long, rod or oval shaped organelles. These are double-walled, provided with an outer and inner membrane. Inner membrane has many infoldings called cristae, which help to increase the surface area for chemical reactions. It contains its own DNA and ribosomes. Inner membrane contains F1 particles or oxysomes which are associated with energy production. Function. Their main function is to manufacture ATP, which is an energy molecule of the cell. ATP stands for adenosine triphosphate, which is energy currency of the cell. Plastids. These organelles are found only in plant cells. They are double membrane organelle which contain their own DNA and ribosomes. On the basis of structure, pigments and functions, plastids are of two types, leucoplast and chromoplast. Leucoplast, these are colorless plastids and they serve to store reserve food material like starch, fats and proteins. Chromoplast, these are colored plastids due to the presence of various pigments. Green pigmented plastids are also known as chloroplast. Chromoplasts also provide color to fruits and leaves. Ribosomes. Ribosomes form the protein synthesizing machine of the cell. These are found in all living cells since these are the site of protein synthesis. Each ribosome consists of a smaller and a larger unit. In prokaryotes, ribosomes are of 70S, smaller unit 30S and larger unit 50S. In eukaryotes, ribosomes are of 80S, smaller unit 40S and larger unit 60S. Here, S stands for Swedberg, a unit used to measure how fast molecules move in a centrifuge. Vacuoles. Structure. These are large single membrane bound bodies which are present in cytoplasm. These are mainly found in plant cells. Their size is also very large and it occupies up to 50 to 90% of the cell volume. In animal cells, their size is very small. Outermost membrane of vacuole is called tonoplast. Functions. Vacuoles mainly store cell sap and provide turgidity and rigidity of the cell. Many important minerals, salts and other soluble substances are stored within the vacuoles. In organisms like amoeba, vacuoles are found as food vacuoles for digestion of food particles. This brings us to the end of the video series on cell. Thank you for viewing. Keep revising. The videos on next topics will be uploaded soon. Till then, like, share and press the bell icon to receive the updates.